A very good evening, respected faculty members and members of the audience. I am Vidushi Gupta, the Journal Club Coordinator of Aspire, and I welcome you all to the ninth Journal Club meeting. Before starting the club, I would like to tell you the flow of events. We will be starting with the pre-club discussion by Shruti Tilak, final year student, to help beginners understand the presentation better. The pre-club discussion will be on the topic Understanding Randomized Control Trials in the first 15 minutes, following which the audience can clear their doubts in the next 5 minutes. This would be followed by two presentations, first by Prashant Saraf, second year, and second by Ansh Agrawal, also second year. Each presentation will be of 20 minutes. Finally, we will be having a post-club discussion of 15 minutes, which will be open for the entire audience and faculty for questions, comments, and feedback. So let's begin with the club. Shruti, you can now take over.
Hi, good afternoon to all. Vidushi, I just wanted to check in real quick. Is my audio okay?
Yes, Shruti.
Awesome. Um, Hello, everyone. My name is Shruti, and I will be kicking off today's Journal Club with a short presentation on understanding randomized control trials. Great. Is my presentation visible?
Yes, Shruti.
Okay, great. So, So, what is a randomized control trial? A randomized control trial is a prospective experimental study that randomly assigns exposures to different groups of participation in within a study population. So, essentially, what we're doing is we have our determined study population, and we're dividing them into groups essentially. And the division into those groups is going to be based on a random allocation. That's a very important part of what an RCT is. And then those respective groups receive different interventions or controls. They basically are assigned an exposure by us, the investigators, right? And then we're going to check what the outcome is for each group and compare the groups. So that's an RCT in a nutshell. I'd like to draw your attention to the position of RCTs on the research or the evidence pyramid and highlight the fact that RCTs are generate a substantially high degree of evidence. By this, what I mean is that compared to a case report or a case study, even a cross-sectional observational study, even a cohort study for that matter, a randomized control trial is something that's superior in terms of the evidence that it generates because the groups that we're studying, the only difference between those two groups, ideally, is the exposure or the intervention that we are studying. So if there is a statistically significant difference between those two groups, then we know for sure that the intervention works or whatever we're studying has an effect. Hence, they're pretty high up on the research or the evidence pyramid. Um, so most of you guys are second years. I hope that the second years are by now familiar with this. There are ideally 4 phases in typically a drug trial, wherein the first phase and the RCTs are started usually after a substantial amount of evidence is gathered lower down in the pyramid. So say you had a lot of case reports getting done on that particular drug and its use. Then someone did a cross-sectional observational study and found some significant statistics. And then someone did maybe a retrospective or a prospective cohort study, analyzed some records, and then found that, wait, we can actually assign this to groups ourselves and see if whether or not it actually does work. So based on all of the early research, maybe animal studies, etc., we go into what is called phase 1, wherein we check the safety and the what dosage is okay for humans to be studied in. And then once we have the phase 1 data, we go into phase 2, which is essentially what we're doing is an RCT, but we're doing it on patients now as compared to in healthy volunteers what we did in phase 1. And then we're doing it in a relatively small population to see how effective it is, gathering some preliminary data. And then if that data is encouraging, then we can move on to phase 3. Now, phase 3 is the major chunk of like well-designed, well-published RCTs, wherein there's a very robust randomization, robust blinding. There are usually multiple groups, and then a lot of extensive analysis is done. And then based on the results of RCT phase 3, you can kind of say that, okay, this drug works. and you can market it, send it out in the community for doctors to prescribe to their patients. And then, sort of, to complete the feedback loop, we have something called phase 4, which is the post marketing surveillance, wherein like thousands of patients are kind of surveilled upon how the medicine is working, whether or not there are adverse events, etc. Anything that we missed essentially in phase 1, 2, and 3 is covered in phase 4, ideally. So this is a general overview of the phases. This is something that we will need to know as part of our pharmacology curriculum as well. Now,
But
there's a lot of...
Shruti, I need one clarification.
Yes,
What is
ma'am.
the difference between phases and device studies and Ayush-based studies? Because what you're
Uh,
talking is only about allopathic drug trials.
Right, ma'am. Ma'am, in the context of phases, we usually kind of correlate them with, I think, drug trials, like new drug development trials. Ma'am, I'm not 100% sure, but I think that for alternative therapies as well, it does kind of go in a similar way, wherein there are
No,
exploratory studies.
no.
Oh, okay.
Actually, when it is a phytopharmaceutical study, so if a phytopharmaceutical is actually defined in our regulatory parlance, that there should be four ingredients in one phytopharmaceutical preparation, that is considered as a new drug. But or
Oh,
else, if there is already a literature on and it has been used from last 20, 30 decades, that is called as Ayush drugs.
I
Such
see.
drugs are directly tried in phase 3. You don't have to do phase 1 and 2. So
Oh,
it is
that...
already in practice. And second is device studies. You don't have to do a phase 1. After preclinical evidence, you directly go to phase 2.
Right,
So you don't, because device studies are never done in normal volunteers.
right, right.
So it directly
Absolutely.
comes to phase 2. So that is
Absolutely.
the only difference in our cities of Ayush, because that is the common, drug trials which are done in India. Device
Right.
studies, Ayush group of drugs, and third or fourth category is FDC, fixed dose combinations. So there are two drugs which are already marketed. The first time you're trying to combine, and then you can directly go on phase 2. And the fifth type of study is called as metronomic study, which is done in oncology, where the drug is actually there in the market and you are giving a subtherapeutic dose for a long time. So that drug is
Absolutely.
already studied for a long time. You directly started phase 3. So these are some sort of differences, and they are all as it is.
Absolutely, ma'am. Thank you, ma'am. I learned something new today. Thank you so much for that. So there's a, as you can see, new drug development is just one sort of scaffold that we learn. There's a lot of variation that goes on depending on the therapy and its sort of status that we're studying. Thank you, ma'am. I'll just move on. So as I was saying, there's a lot of nuance, like ma'am just right now beautifully illustrated. You can imagine there's a lot of nuance that goes into sort of designing an RCT, what we're studying, whom we're studying, and how we're going about it. This is all very important because again, I want to highlight the randomized controlled part of this sort of this study design. We're kind of making sure that it's the most pristine, idealized environment wherein the two groups are really not very different and we're maintaining that in a very robust way. So, in order to ensure that, there's a lot of nuance in terms of what we study. As ma'am just said, there can be drugs, antibiotics, chemotherapeutic drugs, vaccine trials, which is something that's very hot right now. There can be a new diagnostic modality that you're actually checking whether or not it is superior to the ones that exist. Then there can be different surgical methods, different methods of hernia repair. Everything that we learn right now has been previously studied, hopefully with an RCT that determined its superiority and its status. There can be radiation therapies, behavioral interventions, lifestyle modifications, and probiotics. Ayush, as ma'am very rightly pointed out, is one more thing that can be studied under the purview of an RCT. Then the groups. So as I said, the basic point is that we're dividing it into groups and making sure that the groups are as similar as possible. And then the only difference between those groups is whether or not they have received the assigned exposure. Now, in the process of this sort of receiving and allocating the assigned exposure, what happens to the control group? So the control group can be the group that receives a standard of care, that they're not really doing anything other than what they would have done in a clinical setting to those people. That would be standard of care. Then there can be something called placebo controlled, wherein you're not really giving a drug, you're kind of giving something that doesn't have an effect. It's like a null drug, um, a null sort of pill, as compared to the actual study group that is receiving the actual pill. Then there can be active control. Active control again is kind of standard of care, or it can actually test it against another sort of intervention that you want to compare your intervention with. And then Honestly, there can a lot of times there can be multiple arms. So you have like a placebo arm, you have your study group, then you have your placebo control group, and you have an active group. That is also a scenario that you can do. One interesting thing that I want to point out that came about because of certain ethical issues, like how can you just not give anything to the control group, right? How can you just withhold treatment from them, withhold potentially beneficial treatment from them? So because this was an ethical consideration, something called crossover trials came up. Now crossover trials basically have like a switch out between the control and the intervention group, wherein, as you can see in the flowchart, in period one, so first you took group A and then you gave them the actual drug, and you gave group B the placebo. And then you kind of switch them out, wherein now group A is receiving the control and group B is receiving the intervention with the washout period in the middle, so that sort of the residual effects of the drug or whatever intervention you're studying are kind of washed out essentially. Of course, now you can imagine that you can't always do this, but this is an option that can be done successfully in certain situations to solve that ethical sort of dilemma, right? Then there can be different endpoints. Now, this is a little in the domain of statistical analysis that we would do later, but it is important to determine what exactly is the outcome measure, the endpoint that you are studying. That's what's most important to you. What's most important to you, is it how many people you cured, how many people you like kept out of the hospital, how many people you prevented the deaths of, or is it a number? Like, did you reduce the mean blood pressure of your study group by at least 20 millimeters of Hg, something like that. And therefore, you can see that there can be qualitative or quantitative, that is, dichotomous or continuous sort of endpoints that you can check. So this is just kind of an illustration of it. I hope it gives you an overview. Now see, so randomization, as I've been harping up until now, randomization is essentially done to eliminate any confounding factors. The point of this is, if you randomize a study, then you can think about certain confounding factors, like in a case control study, you can match them, but you can always, there's always a margin of error of missing out on certain confounding factors and hence not having two equivalent groups. Therefore, if you randomize, then probability takes care of everything, right? So that's the basic gist of why we randomize. And older methods of randomization do include point toss, random number tables were used. These days, it's all computerized. The computer is fed the data with all of the parameters. It takes care that the two groups are randomly assigned into equivalent groups, right? Now, blinding. So essentially, this mechanism is to sort of prevent something called a selection bias, or even to prevent something called sort of a Hawthorne bias or like an observer bias. Wherein if the patient knows that I am being given the treatment, then it's possible that the patient kind of feels even more like there's a psychological confounding into their sort of treatment outcome. So they feel better than they actually would have if they had not known. Something like that is observed as a psychological effect. So to sort of eliminate that or to reduce that, we have something called blinding. So in a single blinded study, what you understand from the term single blind is that the patient doesn't know whether or not they have been put into the active group or the control group. A double blind study is even better because it will eliminate the sort of observer bias as well. Wherein if I'm an investigator and I know that I have assigned this patient into the study group, then I will kind of also give them more care. Like psychologically, I would give them better health care and therefore their outcomes would be better. And that's a bias, right? That's not an equivalence between the two groups, and we don't want that. Therefore, a double-blinded study is wherein even the investigator doesn't know if that patient is in the active group, is in the intervention group, or if he's in the placebo group or the sort of control group. A triple blinded study is the best, wherein neither the patient knows what group he is in, nor the investigator, nor the assessors. Now, the assessors are the ones that, after the entire thing is done, go into the analysis of it. So even during analysis, that blinding is maintained so that the data is not tampered with. So yeah, that's blinding. Another important part of an RCT that you need to keep an eye out for. This is one thing that I would like to just quickly touch upon. A trial can aim to do different things or check for different things. Now, if you have therapy A and you want to compare that with therapy B, you can have three things that you want to check. Either you want to say, or either you want to establish that therapy A is not inferior to therapy B, or you can say therapy A and therapy B are the same. They are the same in terms of efficacy, for example, or you can say that therapy A is superior to therapy B. So these kinds of trials do exist, and correct me if I'm wrong, ma'am, but I do believe that they have a bearing on the sample size and sort of the quality of the study design in an RCT. So I just wanted to touch upon that.
Yeah, usually superiority designs are usually not done nowadays because we don't have that type of drugs only coming into the market. So
Ah,
maximum studies which are done is non-inferiority studies because you're
oh,
always all are me too drugs. Like for example, I have an innovative drug and there is a generic drug coming in market. So I want to
correct,
prove my drug is not inferior to the drug which is already there in market. So I can't
correct,
show superiority. But for superiority, it doesn't mean like an antihypertensive drug coming in the market only it decreases 1 mm of Hg of blood pressure that becomes superior. Okay, so that doesn't have any clinical significance altogether, even if it is statistically significant. So we
correct.
have to understand, and the margin has to be defined beforehand. So if your
Absolutely.
margin is not means it is defeated, your hypothesis is not tested, or it is not significant the data. So then you will not be able to market that drug. So
Absolutely.
people play safe and doing non-inferiority design, and all bioavailability and bioequivalence studies are all equivalence trials. So there are
Right.
very few superiority studies done now
Okay.
versus used to be done before, like before 1990s.
Okay, ma'am.
So now
Understood.
all the designs are non-inferiority.
That's a great clinical perspective for our students. Um, so yeah, so essentially, I just again want to quickly go over this. You already know this, I'm sure. P-value has been covered quite like extensively. It relates to type 1 error, power relates to type 2 error. I hope you all know it by now. Effect size is something that you kind of guess beforehand. We are hypothesizing that there will be a 20 mm of Hg reduction uh, in the sort of in the intervention group as compared to the study or the control group. So that is one example of the effect size that you are kind of predetermining that, okay, if we get a 20 mm of Hg reduction, that's significant. Um, PICO is sort of a gentle recap. You need to always, in any kind of study, RCT or not, need to have a well-defined research question with good definitions for what population is being studied, what is the intervention that you are giving, what are your control groups and how are you defining them and how are you assigning them, and what are the outcome measures. Extremely important. Now, data analysis, I will not go too much in detail about it. I just want to highlight a few things. Intention to treat analysis versus per protocol analysis. Essentially, an intention to, so to give you some background, in an RCT, a lot of the times, what you kind of planned out beforehand in your protocol may or may not happen absolutely in the same way that you want it to. There can be a significant amount of attrition, for example. There can be an unforeseen adverse event, for example. All of these things can come in the way when you actually start an RCT. So when you do an intention to treat analysis, it is always better because you are reporting all of the sort of data after the RCT in the analysis as it happened, with all of the attrition rates accounted for, all of the other events accounted for. And then per protocol is a slightly inferior type of analysis wherein you are analyzing it as if, okay, this would be the data if we were able to prevent all of the sort of human errors or other attrition from occurring. So in an idealized per protocol scenario, this would be the analysis. That's kind of the difference between the two. Again, key statistics, I will not go into too much detail regarding this, but they are just kind of percentages. Okay, how many, what percentage of our active intervention group were we able to cure or were we able to save the deaths of above and beyond the control group? Something like that. That's relative risk. There is a lot in this. I know the table is kind of a lot to take in, but I will not go into too much detail. Um, Now, rate ratio, again, it's kind of the rate ratio is the incidence of a particular event occurring at whatever the incidence rate the period is in the exposed group versus the non-exposed group. So essentially you're dividing. The rate ratio is the incidence in the exposed group versus or divided by the incidence rate in the unexposed group. A helpful example if you'd like to peruse. Now, survival analysis, another sort of ahead of the two papers that we'll be discussing, I felt the need to just touch upon these. A survival analysis is to illustrate essentially how many men were standing at over a period of time? How many men were standing at 50% of the time interval, at 100% of the time interval? Essentially, how many men were standing could mean, particularly here, classically, survival rates. How many people died versus how many people actually survived over what period of time? You plot it against time. Number of arrests, percentages of adverse events, etc. These can also be sort of done with regards to a survival analysis. And this is kind of the sort of the log curve that you get at the end of a survival analysis. And Kaplan-Meier is a type of analysis that essentially plots a curve and gives you the sort of survival rates. Cox regression is something that you will maybe find that in to be in contrast with Kaplan-Meier. Cox regression is basically just it also accounts for certain multivariates or certain sort of covariates in the particular parameter that you are studying. Nothing to go into too much detail about, just wanted to sort of define. And finally, I know I'm short on time. I apologize to Vidushi. A note on ethics. We do not want to forget that an RCT is a pretty big deal. And in terms of not just sort of the evidence that it hopes to gather, but also in terms of we are the ones assigning exposure. We're essentially playing God. So there are a lot of things to keep in mind. For example, what if this intervention that I am assigning to someone can potentially harm them or lead to an adverse event? What is the data on this? You need to know beforehand going in. What is the level of risk? Then is it a vulnerable group? Is it in pregnant women? Is it in children, etc.? These can be important considerations that sort of give a word of caution to us before we start an RCT. And you, again, like I said, you can't really test one treatment when a clearly superior one exists. You can't test 1 millimeter of Hg sort of efficacy antihypertensive drug when your losartans exist, right? You can't do that. It's kind of an ethical dilemma, and that these are some of the ethical questions that I wanted to highlight. And that's it from my side. I hand over to Vidushi and the audience for any questions.
Shruti, we have a question in the chat box.
Okay, I'll just give me a second. Yes. Kiran, to answer your question, a washout period is basically the interim period wherein sort of the drug treatment slash the control is kind of stopped and it's kind of halted for that particular period of time, just so the residual effects of the drug that you have assigned can wash out. So yeah, you are correct. Is there
Okay,
anything
thank
else?
you. No, no,
Sure,
thank
sure.
you.
If there are any more questions, then please ask.
If not, then that's it from my side. Vidushi, if you can take it ahead.
Okay. Uh, there is another question, Shruti. Please explain survival analysis.
Sure. Akanksha, to kind of bring back survival analysis, essentially, I'd like to illustrate. Suppose that you have a treatment therapy by which you want to reduce the death rate, right? So one way to kind of put up your figures after the analysis or during the analysis period would be to just kind of give you the percentage of death rates in the sort of intervention group versus the control group. Another interesting way to put it is to plot it against time. Your survival analysis is essentially your percentage of survival or the percentage of the participants who are still alive over a period of time. And that number, because the disease I'm guessing that they're suffering from tends to have a death rate, is going to decline, but you're comparing the sort of the slope of the decline between the intervention group versus the control group. And it's great if you're able to slow down that decline or slow down that death rate in your intervention group. That means that your intervention was successful at kind of uh, slowing down that decay in the number of people still alive over time. Does that make sense, Akansha? Please tell me if you understood. Okay, awesome. Prashant, if you'd like to add anything here, because I do believe that survival analysis is something that's covered in your paper.
Yeah, I will cover it during my presentation.
Excellent, great.
So
Um,
in nutshell, what is survival analysis is you do, there are two arms. One is your treatment arm and second may be the standard of care arm. And you started with 30 patients with pancreatic cancer. So as you go ahead in time, like, the 30 will reduce to 29. And at the end of 6 months, there are only 4 patients remaining in standard of care arm and there are 7 patients remaining in test arm. So you actually see, like, at the end of the time period which you have defined, so the time period has to be defined, how many actually survived. So that is called as overall survival. And overall survival is a robust, a parameter which is an endpoint which is actually a good endpoint in oncology studies, because you understand, like, at the end of the day how many have survived. But this is only relevant to the metastatic setting patients, because there, you surely, like, patients are going to die in 6 and 10 months. So your study duration will be not beyond 2 years. But for example, if you want to take a cardiovascular disease and you want to find a survival analysis, it is impossible, because we can't predict when a cardiovascular risk patient may die. So you can't go on years together. So there we do, cardiovascular safety studies or long-term cardiac safety studies, and we see, we find out, we try to presume, like, in 6 months, if you give all this primary prevention, secondary prevention, how many of them are mortality, what is all-cause mortality, some sort of parameter we take in where it is not possible. But or else in oncology study, this is very relevant. That is your overall survival. And one of the parameter by which we analyze this is survival analysis. So another thing where survival analysis is done is when you see toxicity of any drug. So you find out, like, with this toxicity, how many deaths have occurred. So if the toxicity is very severe of any some drug, like, or is like COVID-19 infection, where, the mortality is very high per se, the disease itself is so high mortality, you take all severe patients and then you see survival analysis. So you take disorders where this type of analysis is relevant, and you try to find out, and you see, like, which treatment arm is beneficial to control mortality. That is what it interprets at the end of the day. So it is a type of hazard ratio, and that is the extension of risk ratio in nutshell for
Yes, ma'am.
underdiagnosis.
Thank you, ma'am.
Thank you.
Thank you so much. So moving ahead, um, we extend a warm welcome to Dr. Yashashri Shetty ma'am, who has joined us as the judge for the club. Before starting the presentations, I would like to tell you the rules for the same. Meanwhile, Prashant, you can start sharing your screen. The topic of the presentation, selected only after thorough literature search in the guidance and subsequent approval by the subject mentors of the presenters, should be within the scope of health sciences. Each presentation will be of maximum 20 minutes, followed by a 5-minute questions and feedback round by the faculty. It is a sincere request to all the attendees of the Journal Club to keep their mics off while the presentation is going on. No one is allowed to speak in between. Those having any doubts can wait for the presentation to get over and can ask directly or type in the chat box. We shall now start our first presentation. Over to Prashant.
Yes, Vidushi, can you please confirm if I am clearly audible and the PowerPoint is visible?
Yes, you're audible and it's visible.
Okay, thank you. So, good afternoon, everyone. I am Prashant Saraf. I am second year MBBS student from SET G.S.M.C. And today I am going to present the article Repurposed Antiviral Drugs for COVID-19. This is the interim WHO solidarity trial results. The authors are mentioned in the supplementary appendix, the WHO Solidarity Trial Consortium. I would like to start with what is the meaning of the word repurposed. So the antiviral drugs, they have their indications to treat the particular disease, the particular viral disease. But since the COVID-19 is a new disease, and at that time of the study, we did not know much about what treatments we can give. So some drugs were identified and they were repurposed other than their usual indications to see if they can benefit in COVID-19. So with this background, I would like to proceed. And the main aim of my article today will be to focus on how a randomized control trial is designed and how is it executed. We will not be much dealing with the numbers and the statistical part. So let's continue. This article was published in the New England Journal of Medicine with the impact factor of 91.245 in the year 2020, which is the highest in the category of the general medicine journals. It was published online on 2nd December 2020 and offline as a hard copy on 11th February 2021. And this is a type of randomized control trial. I have referred to the CONSORT 2010 statement, which is a 25-item checklist, and CONSORT stands for Consolidated Standards of Reporting Trials. So they tell us how a randomized control trial should be reported. Important points are the participant flow. That is, a diagram is strongly recommended in the paper, like how the participants were recruited and how the treatment proceeded. And apart from it, the baseline data, that is a table which is showing the demographics and the clinical characteristics of the group at the starting of the trial, so that we get the information if the groups that were assigned, like the control group and the active trial group, if they were having the similar characteristics. Apart from this, I have also referred to the C.S.M.C RCT standard checklist. C.S.M.C stands for the Critical Appraisal uh, Skills Program, and various checklists are available for the different study designs. So I have referred to the RCT checklist. At the end of the presentation, we will see which get all these points. It is a the first point is the PICO that Shruti has mentioned. P stands for the population study, I for the intervention given, C what the comparators were chosen, and O means the outcomes. It is an 11-point checklist. Coming to the background of my article, in early 2020, there were no approved antiviral treatments for COVID. Evaluation of treatments recommended by the WHO Research Forum. So basically, the Global Research Forum and the platform for the research innovation of WHO, it recommended that we should begin randomized control trials for COVID to see if the drugs are helpful and they can reduce the mortality. So, four antiviral drugs were identified and they were repurposed. These include the remdesivir, hydroxychloroquine, lopinavir, interferon beta 1. This is an adaptive trial, drugs that can be adopted and added. So as you will see in the presentation, hydroxychloroquine, lopinavir, and interferon beta 1A were dropped, and the new drugs are added. Currently, the Solidarity Plus trial is going on, where there are the drugs like artesunate, infliximab, and imatinib. And this article reports the interim results for these original 4 drugs. Interim results means that the study is not completed, and the data which is collected during that time, it is analyzed and it is reported. So this trial was registered at both ISRCTN and clinicaltrials.gov. Core protocol was approved by WHO Ethics Committee and also by the national ethics committees and the regulatory authorities. This trial followed the principles of Declaration of Helsinki and GCP guidelines, and it was funded by WHO. Coming to the methods, the trial design is a large, simple, international, open-label randomized trial. So what is the meaning of open label? Open label means that though the trial is randomized, it is not blinded. Means the patients as well as the investigators, that means the doctors who will administer the drugs, they know what drugs they are going to administer. So it is not a blinded trial. This table shows the schedule of assessments and the procedures. It is given in the protocol. So we will see each of these points in detail in the presentation. So starting, like first the patients were judged, like if they are eligible to participate in the study. They were confirmed if they are COVID positive. Informed consent was obtained. So this was at the time of the recruitment. Then the randomization was done, and at this site, patient identity, concomitant conditions, and severity of disease, these all the details, they were uploaded in a cloud-based system. On this study, it did not involve any paperwork. All the procedures were online. And the study then issued a patient study number and a random treatment allocation. That means from the web, it was a web-based randomized allocation. So it let the investigator know what the patient, which group the patient is assigned, which like it is a control group, or he or she will receive remdesivir, hydroxychloroquine, or lopinavir or interferon. Now, during the study intervention, after the patient is allocated, the antiviral drugs are administered, and this is during the hospitalization period. And if any serious or unexpected adverse reactions are there, then they are to be reported using the same website. And while reporting the outcomes, this is at death or hospital discharge. If the patient recovers, then the outcomes will be reported during the hospital discharge, or if the patient dies during the treatment, then at the time of death, the outcomes will be reported. And this included like what study drug was given, if the ventilation was given or not, what was the duration of stay, and if died in the hospital, the cause of death. And a reminder was sent if the reporting was not done within 6 weeks. Coming to eligibility, the inclusion criteria included the patient should be of age more than 18 years and who can give the written consent, who is hospitalized with definite COVID-19, like he is RT-PCR positive for COVID-19, who is not already receiving any of the study drugs, and who has no allergy or contraindications to any of these drugs. And this was in the view of treating physician. The treating physician was given important role to deal with any of the conditions of the patient, like even if he or she has to stop the drugs that are given to the patient, so he can do that because he is responsible for the health of the patient without anticipated transfer within 72 hours to a non-study hospital. That means the patient is expected to stay for at least 3 days in the same hospital, which is included in the study. Exclusion criteria was that there should be, if there is any contraindication to any one of the study drugs, for example, if there is chronic liver disease or heart disease or pregnancy, etc. Then coming to randomization, the randomization was 1:1. That means for every one patient who was allotted to any of the trial drug, one patient was allotted to the control group. And this was unbiased. Unbiased means as this trial was conducted over 6 geographical WHO regions involving more than 30 countries and 400 hospitals. So unbiased allocation or unbiased randomization means the control and the patient who has been allotted to the drug, that is the trial drug group. So they were both from the same hospital or either both from the same countries to eliminate the biases of involving different countries and different hospitals. So there were 5 groups. The control group, all the 5 groups, they received the local standard of care. Apart from this, the group which was allotted remdesivir trial drug, the regimen is also mentioned here. Remdesivir was given intravenously, 200 mg on day 0, 100 mg on days 1 through day 9. So it was for 10 days. For hydroxychloroquine group, 2 loading doses were given and then continued for up to 10 days. For lopinavir, 2 tablets twice daily for 14 days, and interferon beta 1 subcutaneous and intravenous formulations were available. Intravenous formulations were for the patients who were serious, like who were receiving high flow oxygen, ventilation, or ECMO, which means the extracorporeal membrane oxygenation. The article mentions that the 3 drugs, hydroxychloroquine, lopinavir, and interferon beta 1, were stopped, and the dates are mentioned here: June 19, July 4, and October 16. The point that we have to understand by mentioning the regimen is that the drugs, hydroxychloroquine and lopinavir, were given as the tablet, that is the oral formulation. So the patient who had ventilation, who were on ventilation, so they were excluded from receiving these drugs. Coming to the outcomes, the article and protocol both clearly define the primary outcome, which is to assess the effects on in-hospital mortality, that is the death during the original hospitalization. Importance of primary outcome is that the study is designed to answer only the primary outcome. That means the study can best answer only the primary outcome. It can also answer secondary outcomes, but the design was not primarily for it. And so if your concern is about the primary outcome, then this study is good for you. But if your concern is about the secondary outcomes, then you should look for a study which has your outcome as the primary outcome. The secondary outcomes of this study were the initiation of mechanical ventilation, which means if using a drug, it delayed or it reduced the initiation of mechanical ventilation, or it reduced the hospitalization duration. Coming to the sample size, the protocol didn't mention any specific number. It mentioned that the larger the number, more the accuracy will be. because at that time it was not known how the epidemic will develop. Coming to statistical analysis, as you have already now known, intention to treat analysis, which means that you will be assigning every patient who has been randomized if he or she gives the consent. And the primary analysis was of the in-hospital mortality, and the three tests or the three methods that were used are the log rank analysis, Kaplan-Meier methods, Cox proportional hazards model, or the Cox regression. So ma'am has also discussed the Kaplan-Meier and Cox proportional hazards method. The log rank analysis is basically a statistical test which compares two groups, like if the mortality is significantly different in the two groups, which here means the control group and the drug group. So it gives a p-value, and if the p-value is significant, which means there are the two groups, they have the significant difference, and that the drug is either reducing the mortality or increasing. Subgroup analysis were done for the age and the respiratory support at entry. Subgroup analysis means the participants or the study population, they were stratified according to the age groups and respiratory support, like whether they were receiving respiratory support at the entry or not, and the analysis were done separately for. Then coming to the results, so I know this is a very big table, so it might not be clearly visible to you, but I would point out the important points, like 11,330 patients were enrolled from March 22 to October 4, and out of which 64 patients were excluded because they had unknown consent or they had no consent to follow. So as you can see, this table or this flow diagram is given, which is one of the points which is mentioned in consort guidelines. So 11,266 remained in the intention to treat analysis. And there is a partial overlap of each control group with other groups. This means that if in a hospital, more than one drug is available, which can be given to the patients, so one control will be common to more than one trial drug group. That means one patient is receiving control, one patient is receiving remdesivir, and one patient is receiving hydroxychloroquine. So that control, one patient who is the control, is for both remdesivir group as well as hydroxychloroquine group. So as I have told, we would not be focusing on the numbers. We have to see how a randomized control trial is planned and how a flow diagram is given. So these were the patients who were randomized, then this is the remdesivir group. So this number of patients were assigned remdesivir and these were not assigned. So this is the control group. And this number of patients, they did not had consent or unknown consent. Similarly for this, and this distribution is given whether the patients were included or they died or left the hospital and entered the trial before September or not yet reported and still counting. So coming to the baseline characteristics. So this table is also important and also mentioned in the consort guidelines. So the entry characteristics according to the random assignment, which means that we should know like whether the group that was assigned the drug and the control group, whether they were similar at the time of assignment of the drug. Because if they are not similar, then the results might be, it will act as a confounding factor and the results might not be very helpful for us. So we can choose any of these numbers. Like for remdesivir and its control, less than 50 years, 961 patients were there, and for control, for less than 50 years, 952 patients was there. So this is almost similar. And for 50 to 69 years, 1,282 patients, and for control group, 1,287 patients. So the table that includes the total, that is any intention to treat analysis, remdesivir, hydroxychloroquine, lopinavir, and interferon with its control. Also, apart from it, the table has given the baseline characteristics according to the age, as we have seen, and the respiratory support. Like there was no supplemental oxygen at entry, supplemental oxygen, and already receiving ventilation. Apart from it, the other data like lesions in lungs, previous days in hospitals, geographic regions, which includes various countries of Europe, Canada, Latin America, Asia, and Africa are mentioned. Other characteristics like coexisting conditions and adherence to assigned treatments are given. So coming to the results, so this is a graph, the Kaplan-Meier graph of in-hospital mortality at that time. So this is the primary outcome of the graph. So we will not go into detail as how this graph is plotted, but we will focus on the result, like the rate ratio 0.95 and 95% confidence interval, and the range is 0.81 to 1.1. We will understand more in the upcoming slides in the forest plot. So coming to this, like the rate ratio is 0.95. So it may signify that the remdesivir is having slightly less deaths as compared to the controls. But we have to check the p-value also, which is 0.5 by the log rank test. As I mentioned earlier, log rank test is a statistical test of significance, and we are comparing the two groups, like the control and remdesivir. And the p-value is not significant because it is more than 0.05. And it is similar for hydroxychloroquine and its control, for lopinavir and its control. The p-value, the rate ratio is for hydroxychloroquine is more than 1.19, and p-value is not significant. Rate ratio for lopinavir is 1 and p-value 0.97, which is also not significant. And similar is the case for interferon. So now coming to this plot, this is a forest plot, and all the four drugs, remdesivir, hydroxychloroquine, lopinavir, and interferon, are plotted. And as you can see here, the left side arrow, which means the active treatment is better, and the right side arrow means the control is better. So as you move to the left side, it indicates that your treatment, the drug that you are giving, is better. And as you move to the right side, it means the control is better. So I have magnified this for remdesivir and hydroxychloroquine. Coming and the rate ratio for death. These squares and these horizontal lines, they indicate 99% confidence interval, and this diamond, it indicates 95% confidence interval because it is for the total. And for each subgroup, there is 99% confidence interval. So what exactly this means is that, for example, if we take total for remdesivir subgroup, the rate ratio is 0.95 and the confidence interval is 95%, as this is a diamond, and the range is 0.81 to 1.11. So what it means is that if you repeat the trials 100 times, then 95% it is possible that you will get a value which is between 0.81 and 1.11. So even though 0.95 value, it might be significant, like it is less, the remdesivir is causing less deaths than the control group. But when you will repeat the trial 100 times, so there is a possibility then the value can be more than 1. The value can be up to 1.1. So this is the meaning of confidence interval, and even the p-value here is not significant. Similar is the case for uh, hydroxychloroquine for the total. The rate ratio is 1.19 and the range is 0.89 to 1.59. This 1.59 is very high. That means when you repeat the trials in future, you can get the values which are beyond 1. So it is not good for us. As you can see, this is the stratified average for all the subgroups. So it indicates that the control is better. For remdesivir, it still indicates that the treatment is slightly better because the rate is less than. So coming to discussion, the main outcomes were mortality, initiation of ventilation, and hospitalization duration. We have seen the results of the mortality. The results of initiation of ventilation and hospitalization duration were almost same. There was no difference in the drugs, in the group receiving the drugs and the control group. And so these outcomes were not definitely reduced by any trial drug, either overall or any particular subgroup. Now coming to the strengths of this study, the study design is itself the strength, the randomized control trial, as you have seen in the pyramid. It has the large sample size of more than 11,000 participants. The intention to treat analysis, as it is preferred over the per protocol analysis. Subgroup analysis were supplemented along with primary analysis. Reporting was independent of the funders. So this is important. Why? Because the drugs, they are supplied by the pharma companies. And if they have influence in the results of the trials, then it may lead to the publication or it may lead to the dissemination of false results. So this was not the case here. Reporting was independent of them. Participation of both low and high income countries. So this gives the study high external validity, and that the findings of the study, they can be extrapolated. Meta-analysis of trials existing at that time was provided in this study, like the trials like ACTT or the adaptive COVID-19 treatment trial and the discovery or other trials, they were mentioned, but I have not kept this in this presentation because the scope for today is randomized control trials. I would not like to call these as the limitations of this study. So I have written points to ponder instead. Open label design without placebos. So as Shruti also mentioned, like if you know what drug you are receiving or what treatment you are receiving it is likely for both the patient as well as the investigator to create a bias or to create a psychological confounding. But as the article has mentioned, that the primary outcome here was mortality, and it was least likely to be affected with the open label design. Also, they have given one-to-one randomization. Assignment to any active trial treatment delayed the discharge. So, one interesting point that I found in the article was the recovered patients, otherwise fit for the discharge, were kept in the hospital merely to continue their trial treatment, as we have seen in the regimen. Like for remdesivir, it was for 10 days, and for hydroxychloroquine, it was for 12 days, and so on. So even if the patient is fit for discharge, he was kept in the hospital just to continue the trial treatment. So this is ethically questionable because it gives a toll on both the on economic as well as the psychological aspects of the patients. And the study does not mention any remuneration or any funding that is to be given to the patients for their extra stay or for even participating in the study. They only mentioned that in case of any adverse reaction, which is life threatening, they will be given compensation. So exact reasons for stopping the drugs were not mentioned. Like the 3 drugs, they were stopped, but the exact reasons were not mentioned. Only it was written that the drugs were futile, the results were not good, so they were stopped. External statistical analysis for the independent G.S.M.C, which stands for the Data and uh, Security Monitoring Committee, were unseen by the executive group of the WHO. This is a good point, but with 2 exceptions. That is, when the interim results of the lopinavir and hydroxychloroquine were available, then this executive group requested unblinded statistical analysis. So this is also a point which we should keep in mind. And apart from it, the executive group revised this manuscript, the manuscript that was published, and it was drafted only by the WHO trial team and the external statisticians. So now, as we have seen the article, coming to the quick check of the RCT, CSP RCT checklist. So first question is PICO, that is the population they have defined yes. Intervention clearly defined yes. Comparator, the control group was given and outcomes were clearly measured. So yes. And randomization was carried out. And the intention to treat analysis was there. Where the participants blind to intervention? No, the participants were not blind and the investigators were also not blind, but where the people assessing or analyzing the outcomes blinded. So this was not clearly mentioned. In the article, it was just written that the findings, they are not shared with either the funders or the WHO executive group, but the analyzers, where they were blinded or not, this was not mentioned. So we can't tell that. Where the study group similar? Yes, because we have seen the baseline characteristics. And apart from the experimental intervention, which is the drug, did the study group receive same level of care? Yes, because each one of the study group received the local standard of care. Where the effects of intervention reported comprehensively? Yes. We have not gone into details of the statistics, but yes, they are reported. And confidence intervals were reported. And do the benefits of the experimental intervention outweigh the harms and costs? So here, cost-effective analysis was not done, whether the drugs are helpful or not. And so we can't tell this. Can the results be applied to your local population? Yes, because the participants were the local population only, and they have been 1:1 randomized. So this reduces the bias. And would you consider experimental intervention provide greater value to the people in your care? So if the results were good, that this drug is good, then we would definitely say yes. So here we come to the conclusion that the drugs are not good. So the experimental intervention, the drugs are not good means the drugs do not affect the mortality or the other outcomes. So here we would consider not giving the drugs. So yes. So with this, I conclude my presentation. Thank you. And I would like to answer any of your questions.
Thank you so much, Prashant. Ma'am, you can ask your questions.
Yeah, Prashant, I congratulate you. Like you have presented very well, and I think so. I understood the study, and on this study we had a lot of controversy. This study actually came into KM ethics committee also, and it was actually stopped in between because they dropped the arms of hydroxychloroquine and interferon because they did not act well that time. And this was later, like after the interim analysis came into existence. So we had a 5-arm study, but later they dropped these 2 arms. But REC was not ready to approve this study because of a lot of fallacies in this study. That's a part. I want to understand, like, what is the difference between repurposing and repositioning of drug? Now, this is a repurposing. repurposing of drug. What do you mean by repositioning of drug? How do you define that?
I'm exactly not sure.
So, what is repurposing? You can tell us what is repurposing.
Repurposing means the exact meaning. It can go like for the earlier indication for which it was approved. So we are not using any longer for that indication, but we have repurposed or we are using it for any another indication.
No, we
And
are using that because hydroxychloroquine is already is used in rheumatoid arthritis. It's used in SLE.
yes,
It is used in many more autoimmune disorders, isn't it? So repurposing is that already drug has been studied in detail for some indication, and now we feel because so it was not overnight. It came the drugs came as for repurposing. They actually did a there was a well of a test they have done, and they actually did 1,000 drug studies where they gave this antiviral this thing, and these were the drugs which actually qualified for getting into this antiviral this thing. And among that 100 drugs, these are the 4 drugs which were having highest potency as antiviral drug. So they tried for coronavirus. You understand? So it's not abstractly like any drug has been taken for repurposing. So repurposing is the drug is already studied for in length, all the pharmacokinetics, toxicology, or dynamics. Only you are doing a new indication, so it is called as repurposing. And the other way around, repositioning is the drug is already, it was in phase 1, okay, and it got discontinued because of whatever reason. And now we're bringing the same new chemical entity for some indication. So then that drug has to be studied in length as phase 2, phase 3, phase 4, and then it comes in market. So that is the actual difference between repositioning and repurposing.
yes.
Clear? Okay. So what do you mean by adaptive design? Now this study was an adaptive design. What do you mean by that? You
Adaptive
told in
design,
between, but what is
yes.
exactly? Yeah.
Like the drugs, they can be dropped if the interim analysis results if it is causing more harm or it is not causing any benefits. So the drug can be dropped. And new drugs, if they are identified and if they are found suitable to be repurposed, they will be added.
So is it pre-decided in the protocol, or it
Yes.
can be randomly done in between?
No, the names of the drugs is not mentioned in the protocol that which drugs will be added or what. It was just mentioned that the drugs can be added or the
No. Okay. So you are not understanding the concept of adaptive design. Adaptive design means you should pre-decide in your protocol. Like, I'm starting with these 4 drugs. If these 4 drugs don't work, then I'm going to drop this arm. I'm going to add, I will collapse this arm and this drug will continue with some other arm. For example, these are 4-arm studies, isn't it? And there was a
yes,
control arm. And if remdesivir is not working, for example, so what you will write in a protocol is that arm will be collapsed and all the remdesivir patients will go into other 3 arms. Clear? Like hydroxychloroquine or interferon arm, or that is how it is. Or for example, if there is a placebo controlled arm and the risk is actually increasing in placebo arm as compared to there is a lot of benefit in the interventional arm. So what I say is I collapse this arm and the entire arm will go into standard of care arm, so that the patient is not being deprived of the standard of care, which is actually showing benefit. Patient may die also. Now here it is a mortality study, so if
yes,
there is no benefit, means patient is going to die, isn't it?
yes,
So you have to directly take this. So this is called as futility analysis. For mortality, we do a futility analysis, and that is what they have done, interim analysis. So if there are more deaths in one of the arms, you are going to collapse that arm, and you will give them the whatever standard of care which is available. Okay.
okay,
So that is what is adaptive design, and that has to be pre-decided, because if you don't do it pre-decided, then there are a lot of penalties from the regulators, because you can't go on changing. So here there are many things which will change. Sample size will change, arms will change, design will change. Okay, drug intervention will change. There are a lot of things which is going to change. So you have to pre-decide and write in a protocol what you are going to change, and that is exactly the definition of adaptive trials. And all your now the trials which are coming into existence are adaptive trials, because sponsors are saving a lot of money by doing such type of designs. Or else every protocol they have to come with amendment, and they have to freshly put that study for regulators and EC permission, and it will take a lot of time and energy and money also of the pharmaceutical industry. For saving that, they have got this midway, and this is what is working also many times. Okay, what happens when you do interim analysis to the statistics? What happens to when you do a lot of interim analysis?
as
What
far as
happened to the power of the study?
uh,
The power is decreased, isn't it? Isn't it? So you started with the power of 80%, for example, okay, and your P is less than 0.5. and you actually did one interim analysis. So your alpha spending is there when you do one interim analysis. So when you plan more interim analysis, there is a lot of alpha spending. So your power goes on decreasing. So you have to actually take into account how many interim analysis you are going to do, okay, in your protocol, and accordingly you have to calculate your sample size. Okay, here there is no sample size calculation. This is a pragmatic study. Okay, but actually when you do a lot of interim analysis, you have to actually put that in concern in your sample size calculation. Okay.
Okay.
Okay, so what now here they have written like the patients were not having any drugs, means the drugs which were interventional drugs. So what that patients are called as? Such patients are called as what? Who are not on drugs. Whether in the categorization of COVID patients, are they mild, moderate, or severe COVID patients?
Yes.
Does the treatment differ?
Yes, the treatment will first we have to define what do we mean by mild, moderate, or severe. Like, are we categorizing according to the respiratory support? Like if they require ventilation, and if they require ventilation, then if we are calling them as severe or
So there are three this thing, isn't there? Definition for COVID patients: mild, moderate, severe. One is fever, second is your oxygen saturation, and third is your respiratory rate, isn't it? So all these three are going into higher level than the patient is of severe patients, and the standard of treatment is totally different there in severe patients, isn't it? You go monoclonal antibodies there, isn't it? Tocilizumab or itolizumab, because these are the patients who are going to into cytokine storm, and there is 100% chances of mortality. Versus a mild or moderate patient, you can try these drugs which you don't know is going to work or not going to work. So some of the patients will end up into severe, and some of the patient will self-resolve on their own without the drug also working, isn't it? So they have not defined what mild, moderate, and severe, and what is the standard of care everywhere. Like for example, if you see that hydroxychloroquine dose, what is the guideline in India? Is that dose recommended in India? The dose which is given in this paper?
no, no, ma'am. Here they have chosen the dose for the amoebic liver abscess, the dose of hydroxychloroquine, and not the dose that is given for malaria.
So no, for COVID, what is the guideline given by ICMR? What should be the dose of hydroxychloroquine given in Indian patients? See, because India was one of the sites, isn't it? So I'm
Yes.
asking India-specific questions also. So is the drug dose same which was prescribed in Indian population?
No, ma'am, I'm not
No,
aware of
na.
it.
See, this is the malaria dose they are given. 4 tablets, then 4 tablets 6 hourly, then 2 tablets after 12 hours, and 2 tablets after. So that is a malarial dose they are talking about, but that is not the dose which is recommended, isn't it? For us, the recommended dose is 400 milligram stat, okay, for 2 days, and then it's downgraded for 200 milligram, because hydroxychloroquine is also not free of any toxicity. There are a lot of problems with hydroxychloroquine, isn't it? And somewhere they have mentioned about lopinavir given along with ritonavir. Did you miss that?
No, ma'am.
Somewhere they have written.
Yes, ma'am, they have mentioned, but I have not included to just reduce the complexity. They have mentioned that
Okay.
lopinavir was given with ritonavir to decrease the hepatic degradation, and they have also mentioned up to July 4, the comparator groups were like interferon was given along with lopinavir, and the control group was only the patients who were receiving lopinavir. So the interferon plus lopinavir was compared with lopinavir. It was not compared with direct standard of care group who was not receiving any other trial drug. But after July 4, they considered they have given interferon 1 beta 1A directly, individually without lopinavir, and compared it with the standard of care.
So, what is the take-home message? This one, Prashant, when you saw this study, what you felt like? Was this design very attractive for you to do a study in a clinical practice when you do studies because this study design is such totally different design, which actually is not seen in our cities. Any standard of care, which is available in country, you add one drug in addition. So such protocols are never approved until now. I'll tell you for your information. This was the first protocol which has been designed like this, and this is because only for COVID pandemic. So you have to understand that this type of protocol are only approved in epidemic setting. In routinely, we will not approve this, because if you see the 4 drugs which are being compared, they are totally different drugs. Okay, and the class and the risk, everything is different. So when you do this benefit analysis in all these 5 set of patients in 5 group, as an analyst also, as a pharmacologist, I will be very much skeptical of doing a risk benefit analysis, because there is a lot of data which I don't know how I should compare with, because every molecule is a different molecule. Hydroxychloroquine different class, Lopinavir is a different class, interferon is a different class, remdesivir itself. So all the drugs which have been put did not have market as such, and they were being tried to be improved in this class of COVID, but they did not work. So many times you try to push something, but it doesn't work. That is another take home message when you try to do something. And at that dilemma period, every prescriber was giving this drug, and there was a lot of shortage of these drugs, isn't it? Because there were very few manufacturers in the country, and they were in a view, the demand was created, artificial demand, and the patient did not know like what he's getting and what he's surviving out of. He may be surviving on his own without any drug drug class working there. So you have to understand, and there is a lot of ethical things in this. So as a clinician, you should know like there are so many unethical things happened at that time phase, and hospitals have made a lot of money out of patients' death beds, like patient did not recover at all, and we don't know. And as an expert also, I was also in many panels, and we used to argue a lot, like if these drugs are not going to work. For example, for hydroxychloroquine, we have published 3-4 studies also from molecular basis also how this is going to work. Prophylactically, we were giving in employees, we were saying it is very challenging, to give this drug, which is not non-toxic. It is toxic. It has eye disorder, eye complications. And if I'm a myopic already and I'm taking hydroxychloroquine, I don't know what is the long-term complications for me also. So we have a lot of reservations, like, very small one example I'm giving you like this. So you as a, practitioner tomorrow, you have to understand when you're reading evidence from such studies, these are the eye-opener studies, and these are very exceptionally approved in situations like, these disasters and in emergencies. And or else such type of protocols are never accepted. And that is how is the take-home message. That is how I feel. You can add something like, it was very interesting. And Prashant, at a level of second year MBBS, in your place, I would have not done such a good job. And it's very commendable. And thank you very much.
Thank you. I also got to learn
Yeah.
various new points.
Yeah. And crowd, if you have any questions, you can ask him. I have finished with my questions. Okay, Kiran has one question. An open label design without any blinding. Okay, what is the you can read, please.
Yeah. Question is, an open design without any blinding says that everyone should know the whole treatment procedure. After that, we have various degrees of blinding. But if someone has to give a written consent, then don't you think that the person should know what will be happening with him or her throughout the course of treatment or analysis? Then blinding won't be possible anyways. Any comments? So, as far as I know, it is mentioned in the consent itself that if you are enrolled in the study, then you can be either allotted to the control group or the trial group. So that point is explained while administering the consent. And if the patient agrees to participate, then only he or she is recruited in the trial. Ma'am, if you would like to add something.
Yeah, it is open label means the patient knows that he's in which arm and he can select the arm. So like, for example, I say like, there are 4 drugs which are available, and, these are the 4 drug arms, like, which we are going to put on, and patient may say like this. So many times it is patient preference arm also when it is open label. The thing is, there can be clinician bias here. Okay. So clinician can put a patient of mild disorder to hydroxychloroquine versus a patient who is a severe patient into remdesivir. So you can't actually eliminate investigator bias here, and that is an element of concern for us. So open label actually comes in evidence at a lower grade, but there is randomization, so it comes in that randomized control trial. That much you have to understand. So when you're reading evidence, you have to read with that light of care. Like it is open label, so there can be an investigator bias in that.
Thank you so much, ma'am. Thank you, Prashant. Moving ahead, Ansh, can you present your screen?
Yeah, sure. Just give me a second. Hello. Is my screen visible?
Yes.
And the audio and the video is clear?
Yeah, it's clear.
All right, then I'll start. Good evening to everyone. I'm Ansh Agrawal, a second-year MBBS student at G.S.M.C Medical College and K.M. Hospital, Mumbai. And today I'll be presenting the study Tenofovir versus Placebo to prevent perinatal transmission of Hepatitis B. This is a study by G. Jordan et al. And it was published in the New England Journal of Medicine in the March 2018. So moving on. So, introduction. So, mother to child transmission of hepatitis B virus accounts for the majority of cases of chronic HBV infection and is a leading cause of cirrhosis and hepatocellular carcinoma worldwide. So, without immunization, 30 to 42% of the infants born to HBV infected mothers can develop the infection, and out of this, chronic infection develops in 65 to 90% of infected infants. Universal immunization against HBV, starting at birth with the administration of at least 3 doses of vaccine by 6 months of age, has reduced the prevalence of infection, as well as the additional precaution of administering hepatitis B immunoglobulin at birth to infants born to HBV infected mothers. However, mother to child transmission still occurs in infants born to women with a high HBV viral load, that is greater than 200,000 international units per milliliter, or with HBe AG. Also, HBV escape mutants to hepatitis B vaccine or immunoglobulin have also been detected. So basically, such like the virus undergoes mutations such that even if you give vaccine or immunoglobulin, it is not very effective. So antiviral agents have been found that inhibit HBV replication, such as lamivudine, tenofovir, disproxil fumarate, and telbuvudine. So this may also reduce the risk in mother-to-child transmission. So there was a previous RCT done before this study that was conducted in China, which showed significantly lower rate of mother-to-child HBV transmission among mothers who received TDF than among those who received standard care. But it had two big limitations. It wasn't placebo-controlled and was not double-blind. So there was a need to have a study that overcome these limitations and had a much more robust study design. So what was the aim of the study that we are discussing today? To assess the efficacy and safety of tenofovir disproxil fumarate in preventing mother-to-child HBV transmission in pregnant women with HBeAg positive status. So coming to the methodology, regarding the study design, this was a phase 3 trial. It was multi-center trial. It was conducted in 17 hospitals in Thailand. It was randomized, double-blind, placebo-controlled, parallel group, and clinical trial, which is kind of self-implied from the name RCT. So just one thing, parallel group, as Shruti has also quite wonderfully explained, just wanted to elaborate. Parallel group is basically that if there are two different groups, in this case it is TDF and placebo, from the beginning of the trial till the end, the participants will stay in the same group and will receive the same drug, whether it be TDF or placebo, and the analysis will be calculated accordingly. Regarding the inclusion criteria, women at 28 weeks of gestational age within a window of plus minus 10 days were included. They had to be older than or at least equal to 18 years of age, had to have positive HBsAg and HBeAg status. Also, the ALT level, that is the alanine aminotransferase level of 30 IU or less per liter, should be there at screening, and a level of 60 IU or less per liter at the trial entry. And coming to the exclusion criteria, so the exclusion criteria were positive serologic test for HIV or hepatitis C virus, whether they had received TDF at any other point of time or had received any other anti-HBV treatment during the current pregnancy. The investigators also excluded people who had creatinine clearance of less than 50 ml per minute, confirmed proteinuria, and confirmed normoglycemic glycosyluriah, as well as evidence of a fetal anomaly that was incompatible with life in a pregnant mother in a fetus. Coming to the sampling and participant recruitment, after written informed consent was taken from January 2013 to August 2015, participants were randomly assigned to receive either 300 milligram of TDF or placebo, and then they were placed in two groups in a 1:1 ratio and administered once the TDF was administered, or the TDF or the placebo was administered once daily from 28 weeks of gestation to 2 months postpartum. So we have like a very clear time period where they have given the drug. But I would like to just highlight one point here, that there is no fix, like there is not a very broad consensus on when to give antiviral treatment in cases like these. We'll look at it further when we come, but just a heads up. Regarding the randomization, they use a computer software in which permitted blocks were done, and then it was stratified according to trial site. So in a very basic sense, I won't go too much into depth, but permitted blocks basically means that there are blocks of people, let's say blocks of 4 people, for example, and each block will contain 4 people, and these blocks will be randomly assigned. So 4 random participants will be assigned, whereas the computer will make sure that if 2 blocks are assigned, so let's say 4 out of 4, 2 will go into this and other class. So the computer will make sure that the rest of the characteristics are similar, and that will account for the confounding. And then afterwards it was stratified according to trial site. So stratification is nothing but just basically making subcategories or subgroups in a population, and this according to a defining characteristic. So for this, our characteristic was trial site. Then coming to the sample size, 331 women from 17 public hospitals were taken. In the Tenofovir group, 168 women were placed, and in the placebo group, 163 women were placed. Now you might be wondering, how did they come upon with this sample size? So what the researchers did was that they ran a Fisher's analysis, and they looked at the past tests, which were past trials which were done, and they calculated that if I wanted to conduct a trial with 90% power to detect difference of at least 9 percentage points in the transmission rate. So the researchers expected that there would be, let's say, 3% infection rate in the TDF group in the infants and 12% in the placebo group in the infants. So if all of the parameters are met, so I would need at least 328 women. So that's why they choose 331 women, which is approximately close to it. So this is the participant flow. It's as Prashant mentioned, it is important to look at it. However, I won't be going to much depth. I just hope to convey to you that what happened here. So 2,512 women were assessed for eligibility. Out of this, 2,181 excluded. So they had multiples of reasons, such as tested negative for HBAG, had ALT levels of greater than 30 IU, or did not provide consent, or had other reasons which were mentioned in the exclusion criteria, such as, let's say, prior HIV infection or hepatitis C infection. So 331 underwent randomization, out of which 168 were assigned to and received TDF, 163 were assigned to and received placebo. Out of this, further attrition occurred. So some people were lost to follow-up or withdrew. Some had, let's say, one had a stillbirth in this case, then had infants who were lost to follow-up, or had infants who were withdrawn. So the mothers weren't willing to make the infants be involved in the study. So after all of these attrition factors and all, 147 were included in analysis for primary endpoint in both the groups. So this 147 when they're talking about, I'm talking about the number of infants here, but it also applies to the mothers because it was like a mother-infant pair. So like one mother and one infant. There was a case of twins, but that was only one pair, and we'll look at it, how the researchers handled that. So before we do that, we'll just look up at the follow-up of the mother and child and the schedule which they followed. So maternal visits were at 28, 32, and 36 weeks of gestation, and then at delivery, and at the 1, 2, 3, 4, 6, and 12 months postpartum. Then all the infants had received hepatitis B immunoglobulin at birth and hepatitis B vaccine at birth and at 1 to 4, 6 months through the Thai immunization program, which is the national program which Thailand follows. Infants were breastfed, and infants had blood samples obtained for the determination of HBV infection status before vaccination and at 1, 2, 4, 6, 9, and 12 months of age. So coming to the endpoints, very briefly, endpoints are nothing but the outcomes which a trial is looking at, or the you could say the main thing that the trial wants to look at or analyze or predict or have data for. So the primary efficacy endpoint in our trial was infants' HBV infection status at 6 months of age. So this was done by whether if an infant could have been said to have an HBV infection if he or she had HBsAg positive status, and which was confirmed by the detection of HBV DNA in the infant. So infants with the HBV infection status available at 6 months were included if their mother received at least one dose of TDF or placebo, and they were included in the analysis according to the randomized group assignment. Intention to treat analysis was used. Basically, if let's say some people discontinued the placebo or the TDF regimen, they were placed in that group only till the end of the trial. And where actually the more better way to say were analyzed according to if they were placed in that group till the end of the trial and were not shifted to another group. Coming to the some few other endpoints. So another thing would be safety endpoints. So occurrence of maternal or infant adverse events, one of the safety endpoints which the trial wanted to look at. So in this, serious adverse and grade 3 or 4 signs and symptoms. So what do I mean by grade 3 or 4 signs and symptoms? So there was a division of AIDS table which the researchers have reference to, in which has been classification of grades of the adverse symptoms. So grade 3 is severe and grade 4 is life-threatening. Then after that, maternal hepatic flares, that is ALT levels of greater than 300 IU per liter after the discontinuation of the trial regimen. And lastly, infant growth as measured by the WHO Z scores for age, for weight, height, and head circumference at 6 months. There was also an exploratory endpoint which measured in which maternal HBV DNA level at delivery was measured, provided that the anti-hepatitis B antibody titer of the infants were at least 10 IU per liter at the 6 months of time period. Coming to the statistical analysis used, there were 3 types of tests used here. First one was the Fisher's exact test. So Fisher's exact test was used for the analysis of the efficacy endpoints, adverse events, and hepatic flares. So you might be thinking, why Fisher's? So basically, Fisher's is used for categorical data. So let's say a yes no data would be a very good example. So efficacy, if we talk about efficacy, either the drug is efficacious or not, either it prevents infection or not. So yes, no. Adverse event, is there an adverse event or not? Hepatic flare, is there a hepatic flare or not? So for values such as which have a yes no, you could say you can use Fisher's exact test. Then unpaired t-test is used for the analysis of HBV DNA level and infant growth. So basically unpaired t-test compares the means of two continuous data sets. So if there is numerical value, so what will happen is that unpaired t-test will compare those two and give you whether there is a significant difference or an association. So this was used for the analysis of HBV DNA level and infant growth. Finally, a log rank test was also used. I won't go into much depth about this because it was not very significant results, and it will eat up too much time. But basically, they did it for the time to event analysis of the first adverse event. So they wanted to check whether there was any significant difference in the time period where the first adverse event was reported in the TDF group and in the placebo group. Now, proportions are presented along with 95% exact binomial confidence intervals. So coming to the results. This is quite a big table, and I won't waste much of your time by going through the entirety of it. Some just few points I wanted to highlight. First things, if you look at the TDF group and the placebo group, you will find that they have very similar characteristics. Like, let's talk about age. So median age for TDF group is 25.5, placebo group is 26.7. Then the gestational age is 28.3, 28.1. So basically, this is one of the beauty of randomization, that we have like very similar groups, and this eliminates the risk of bias. Then I would like to drag your attention to the HBV DNA levels. So if you look at the mean HBV DNA levels expressed in log 10 IUs per ml ska value, so you will find that in TDF group 7.6 was the mean value, plus minus 1.5 the standard deviation, and in the placebo group it was 7.3 plus minus 1.7. Please note that this value was at when for the women at baseline, that is when they enrolled in the trial. Another thing that I would like to drag attention to is if you look at the number of women who had greater than 200,000 IU per ml of HBV DNA levels, you could see that in the TDF group at baseline it was 152, and at placebo group it is 142. Also, please note that in the brackets the values which are written. So let's say 90, that is the percentage. So 152 as in 90% of the TDF group had greater than 200,000 values, and 142 as in 87% of the placebo group had greater than 200,000 values. So coming to the delivery, we had number of women with data as 162 and 160. One more thing I would like to drag your attention to is the type of delivery. This will come important later when we are discussing it in the discussion section. But if you would notice that there is a much higher percentage of vaginal deliveries over cesarean section. So if you see 77%, 23% in the TDF group, as compared to the 71% and 29% in the placebo group. So vaginal deliveries were in the majority. Lastly, in this page, please look at the HBV DNA levels, that is of the women on the delivery time, and you will see that there is a drastic difference between in the TDF group and in the placebo group. So in the placebo TDF group, 12% have greater than 200,000 IU per ml HBV levels, and in the placebo group, it's 90% who still have greater than 200,000 IU levels. Coming to the infants, if we talk about infants, we had infants, 163 infants in TDF group with data and 160 in the placebo group, but we didn't analyze every one of them due to various reasons such as dropping out or having incomplete data in between. So just a few things here that I would like you to look at. So hepatitis B immunoglobulin administered. So if you see that there's a very curious case of no, and then there are two infants who haven't been administered immunoglobulin in a TDF group and one in placebo group. So this is due to a multitude of factors. For example, one of these was a stillbirth, and the other one was due to just a second. Vidushi, is my feed still visible?
Yes.
Just give me a minute, okay? Apologies for the break. There was a problem in the line here. Moving on. So if you look at the hepatitis B immunoglobulin levels administered, you will see that there were two infants who weren't administered it in the TDF group and one in the placebo group. So this was due to a combination of factors such as one of the stillbirth and two of the mothers' consent were against hepatitis B immunoglobulin administration. One more thing that I would like you to note is HPV vaccine administered at birth. So you'll see that in the no category, there's one in the placebo group, and this was due to post delivery complications where the infant died in a few hours. So that is why. Another thing you will please note time from birth to HPV vaccine. This is very important because it will come up in the discussion sections later. So if you look at the median value, it's 1.2 hours for both of them. So from birth, within 1.2 hours, HPV vaccine was given. There's the 0.28 to 2.0 range. So between that, so the lowest time was 0.8 and the highest was 2.0. Moving on to the next page. This is the results section or the endpoint section, which is the arguably the most important part of the presentation that I'm going to talk about. So if you please look at the efficacy endpoints in infants at 6 months in the HBV infection percentage in the primary analysis, you will see that in number of participants are same for 147 in both the group. That is, number of infants are same, and there are 0 events in TDF group. That means that there was no hepatitis B infection in infants in the TDF group. So that is a 0 percentage value. The value which you're looking at here is nothing but 0, which is the percentage, and the terms in the bracket are the confidence interval. So 0 to 2. So that means that if I repeated this experiment quite a number of times, so I would be 95% sure that I'll get the values ranging from 0 to 2 only. So as compared to the placebo group, you will see that there are a number of participants of 147, but the number of events are 3, which is a value of 2%. What I want to draw your attention to is the p-value. So even if but just numerically there is a difference, the p-value is 0.12. That is greater than 0.05. Hence, we can say that there is no significant statistical difference. That means that our primary endpoint is has not been proven, and the null hypothesis has been proven that there is no difference between the TDF group and the placebo group. Coming to the safety endpoints, if you look at, so in the hepatic flares, that is the ALT greater than 300 IU per liter, 9 women faced it in the TDF group and 5 in the placebo group with 6 and 3% respectively. The p-value was 0.29, again not significant. Similarly, WHO Z scores were not significant, and adverse events of grade 3 or 4 serious adverse events were not significantly different between the two populations. Just one last thing that I can say is significant is the exploratory endpoint, that is the HBV DNA level among women at delivery, which is 4.0 plus minus 1.6 standard deviation for the TDF group, and for the placebo group, it's 7.3 plus minus 1.7. So as you can see, the p-value is less than 0.001. So this is statistically significant. However, it doesn't contribute much to the study because it is already well established that antivirals will reduce the viral load in a patient. So it is not a very novel finding here. Coming to discussion, a primary endpoint did not reach statistical significance, and thus the trial is considered to be a negative one. That is, the trial did not prove anything new, and in fact reinforced the null hypothesis that there is no difference between TDF and placebo group. One of the reasons I wanted to present this study was that it's not necessary that each time the trial must have a positive finding. Even a negative finding can contribute to scientific literature. So one more thing that we can observe is that a low rate of transmission with an antiviral agent may relate to the reduction in maternal viral load, as we saw in the HBV levels, as well as to pre-exposure prophylaxis for the fetus. The appropriate duration of antiviral treatment is unknown. As I already mentioned, there is no very broad consensus regarding the country's health organizations in what should be the appropriate antiviral treatment duration to be given to pregnant women to prevent HBV infection in the infants. In the trial, one interesting thing to note is that the HBV DNA level of more than 200,000 IU per milliliter was observed at delivery in 12% of the women in TDF group after 10.7 weeks, which is somewhat less than the 25% rate that was observed among women who received TDF for a mean duration of 8.6 weeks in an earlier study, which you can see that I have referenced in below. Also, percentage of HBV infected infants in the placebo group was 2%, which was lower than the originally assumed 12% on the basis of published data, mostly from China. Now, this discrepancy may be due to sampling variations, as in it's a different sample per population, so there inherently there will be some variations in the results. However, it could also be related to the duration between birth and the first administration of the hepatitis B vaccine. There have been some studies which show that it is the duration between or the time interval between birth and hepatitis B vaccine administration is very important. So here it was the mean time was 1.2 hours, so it was going quite early. So that is why one of the reasons that there is low transmission rates. Maternal use of TDF may prevent transmissions that would occur when the birth dose of the vaccine is delayed, but its exact timing has not been reported consistently in previous perinatal studies, and so we can't comment on it. One more thing, amniocentesis and mode of delivery may also be associated with the risk of HBV transmission, according to a study which I have referenced in below. In a trial, 26% of deliveries were cesarean sections, none of which were elective, and no women underwent amniocentesis. Contrast that to the trial in China, where amniocentesis may have been more frequent, and the rate of cesarean sections, which was 50%, was higher than in the trial. So, and also circulating escape mutants in China is higher, which increases the chance of vaccination failure. So these are some of the reasons that the authors have given that why possibly the China trials had showed higher infection rates, as well as a statistically significant difference in TDF and placebo group. Regarding coming to the safety concerns, there was no significant safety concerns associated with TDF in the pregnant women and infants, which is also relevant not only for HPV but also HIV, as TDF is also given as a treatment regimen for HIV. So this can ensure that TDF is safe for HIV treatment in a pregnant woman. Time to first adverse event didn't differ significantly between the two groups. So this is basically the long rank test coming into play. So not much to talk about here. Just it's this much, that there was no significant difference between when the first adverse event appeared in the placebo group and the TDF group in both the women and the infants. As you can see, the p values are 0.5 and 0.62, which are not significant. Infant growth did not differ significantly between groups, although the infants in the TDF group had somewhat lower mean weight for age Z scores at 6 months than those in the placebo group. But again, as it was not a statistically significant difference, we shouldn't comment on this much. Now coming to limitations, the simplest limitation to understand is that the sample size of the study itself was small. 331 participants are not quite a lot when we look at the sizes for RCTs, which can range into the thousands. And even if we talk about a phase 3 trial, so even 700, 800 participants have been known to be recruited in phase 3 trials. Then limitation of recent perinatal HPV infection prevention studies has been the assumptions. So as we saw in the beginning, that the authors assume that there would be a, let's say, 3% infectivity rate in the TDF group and 12% in the placebo group. So this was a flawed assumption, because the real world data was quite different from it. So to rectify that, a superior trial could be done, assessing a difference of 1.9 percentage points. That is, assuming that 0.1% would be infection rate in TDF and 2.0 would be in placebo, with 90% power. But it would require a sample of more than 1,600 mother-infant pairs. So this is not very feasible, as the antiviral treatment cost will increase. So coming to the conclusion. In conclusion, in the context of a low rate of HBV transmission, this trial did not show that maternal use of TDF, in addition to the administration of hepatitis B immunoglobulin and vaccine to infants, resulted in a lower rate of mother-to-child HBV transmission than placebo. So coming to the critical appraisal, there's one more point, like I'm sure, which must have popped in some of your mind, because according to WHO guidelines, we do give TDF a drug in pregnant hepatitis B infected mothers. So why am I talking about this trial? So there's one more point I would like to highlight in the CASP checklist. So I performed my critical appraisal using the CASP checklist. So the new modified version of CASP checklist, which is a 2020 version, I used that, and in it they have also taken into account the consort guidelines. So let's go to the first question. Did the study address a clearly focused research question? Definitely yes. There was a clearly addressed population, as Prashant said, PICO. So population, intervention, comparator, outcome. There was very clearly defined that. Was the assignments of participants to interventions randomized? Definitely yes. We saw permuted blocks with stratification was used using a computer software. Well, all participants who entered the study accounted for at its conclusion, yes. As you saw in the population flow table, quite exhaustively, they have mentioned the reasons for the dropping out of participants, such as losing follow-up or withdrawing consent or some other thing. Was blinding done for participants, investigators, and once analyzing the outcome? So they have mentioned double blinding. So participants blinding was done, investigators blinding was done, but they haven't mentioned analyzing the blinding of the people who are analyzing the outcome. And as they mentioned a double blinded trial, I would like to think that the third category would be no. The rest of two categories are yes. Were the study group similar at the start of the RCT? Yes, as I illustrated that the characteristics were quite similar, so that was definitely there. That is whether treated equally. Yes, definitely, because the standardized protocol was there for both of the participant groups. They were given the hepatitis B immunoglobulin, and vaccination schedule was the same for both of them. Coming to the next point, whether effects of intervention reported comprehensively, yes, quite exhaustively. The effects of intervention were reported. P-values were mentioned, and the tests were mentioned. The tables and graphs were drawn. Was the precision of the estimate of the intervention or treatment effect reported? So this is basically talking about confidence interval. So yes, they have tried to include confidence interval wherever possible. So they have definitely done that. So do the benefits of the experimental intervention outweigh the harms and costs? So I would say no, because in this case, the trial has proved that the TDF does not have any overt benefits over placebo. So in this case, the benefits of the experimental intervention outweighs the harm or cost. I wouldn't say that. Can the results be applied to a local population in a context? I am saying no. There is a bit of a explanation behind this, which I would like to elaborate in the next slide. Before that, we'll continue to the last point. Would the experimental intervention provide greater value to the people in your care than any of the existing interventions? Again, this would be a no, because the experimental intervention, that is TDF, was proven to be less effective. So I would say on the basis of this study trial, I would say no. However, when you talk about the results, can the results be applied to local population? So the study was done in Thailand with no similar RCT in India. Even then, due to the robust randomization techniques used, I would say, okay, if it was still only till this much, I would think of applying the results even there too. But there have been numerous meta-analysis done after 2018, and a meta-analysis done in 2020, the largest of its kind regarding this topic, has shown results contradictory to this trial. The title of the meta-analysis is Efficacy and Safety of Antiviral Prophylaxis During Pregnancy to Prevent Mother-to-Child Transmission of Hepatitis B Virus, a systematic review and meta-analysis. And this meta-analysis concluded that it was in fact favorable, or there was a significant difference when antiviral drugs were given. So it included TDF, it included lamivudine, and so multiple antivirals were there, but the systematic review was very robustly done. And from the findings of this meta-analysis, WHO has made a recommendation for administration of TDF 300 milligram starting from week 28 of pregnancy until at least birth. And this, you can see this on the WHO website as well. So another point that I would like to say here is that sometimes it's not necessary that a study is right or wrong. And in this case, it's not accurate to say right and wrong because it was a sample size or whatever. Maybe it's because of the small sample size or not. But it is important to base your knowledge on the not only on just one study, but on multiple studies. So a meta-analysis proved that the finding that the study had, it contradicted it. So the main take-home message that I would like to say from my presentation would be, number one is that even if it's a negative trial, that means that it proves a null hypothesis, or it shows that there is no significant difference. It doesn't mean that it is useless, because this was published in NEJM, and NEJM is a very good journal. The second point I would like to say is that regarding that it's important to read all of the relevant literature and do literature search. Because if I just read this trial, I would have concluded, okay, TDF does not work. But as a clinician, that would be my mistake. So it's better that I read all the systematic reviews and meta-analysis and more studies done after that, so that I can come to a more correct clinical discussion. That is all. Thank you very much for your time.
So I'll give an option to all the crowd. If anybody has any questions, then I'll start my asking my question. Any crowd, do you have any questions, students? Okay, Ansh, you've done a good job. And
Thank you so much.
studies in pregnancies are very rare. And what are the conditions where studies are actually done in pregnancy? Because if you see, or else in clinical trials, exclusion criteria is all pregnant women, isn't it?
Yes, ma'am. So, ma'am, what I would say is that for studies in pregnancies, perinatal conditions are important. So, like in this, there was a vertical transmission of hepatitis B virus from mother to child. So, in let's say similar infections of sort, for example, we know that the HIV has a tendency of passing on infections from mother to child. So one of these could be where the risk of infection is from mother to child. Then can also be some other perinatal conditions. So mostly stuff that focuses on the diseases related to pregnant women, because if I want to study diseases related to pregnant women, I will have to take a study population which includes pregnant women.
So one thing you said is right, like, this condition is pertaining to pregnant women, so you have to do a study in pregnant women. Now, another thing is, what is the seriousness of the disease? If you don't treat a female with hepatitis B, what happens? What is the outcome? For the maternal side as well as for perinatal or neonate and fetus as well? So what is the
So,
outcome?
ma'am, so if we talk about hepatitis B, so it has like two types of things. Now, it could either be acute or chronic. So, if it's acute, it will resolve on itself. But chronic, so there is possibility that it can move on to hepatocellular carcinoma could be there, ma'am. Or
No, but in
if
pregnancy, you're not thinking of that, isn't it? That can be treated
okay.
after the pregnancy event, isn't it? So now we are
Hmm.
treating in pregnancy as a setting. So what is the risk here in pregnancy? See, when you're doing a study in pregnant women, there are many things you have to think about. One is what is the seriousness of disease? If untreated, what can happen? What are the availability of alternatives available? Because there is already lamivudine and other drugs available. Why I want to try tenofovir? Secondly, when you're doing in pre-marketing setting or post-marketing setting, now tenofovir is already marketed for HIV and some other disorders, and now you are trying a new indication for that. So, what are the considerations which are done in pregnancy?
Ma'am, so not very sure about this, but maybe it could be efficacy. For example, WHO has selectively said that tenofovir, like I went on the WHO website and they have mentioned tenofovir, but the systematic review which I discussed in the end, they've also analyzed lamivudine and other drugs. So maybe if the efficacy is a bit better, but again, ma'am, as you said, we don't normally do superior trials, ma'am. So I'm
Yeah.
not really sure about this one, but efficacy is one thing which is coming to mind. Because even I understand that, ma'am, ki if there is pregnant women, so it is very risky, and unless the trial is absolutely needed, which in this case it was, so that is the only indication of doing the study, ma'am.
So another thing, like in pre-marketing setting and in post-marketing setting, what is the difference? So what is the background data is required before you try to do a study in pregnant women?
Ma'am, if it's pre-marketing, ma'am, I would say that uh, has is there extensive literature? That is actually much more general. But if there is literature before this, and if the literature is if even somewhat good value, I would refrain from doing the study. But ma'am, the reason for doing this study was that before this, only one study was conducted in China, and as I mentioned, it wasn't placebo-controlled, double-blinded. So there was a very acute need of conducting this study. And as I said, the statistics that we have for hepatitis B virus, it's quite significant because if we say that 30 to 42% of hepatitis B infections are transmitted vertically, and out of those 60 to 90% result in chronic infections. So that is a very huge impact on quality of life, ma'am. Not only if you think about mortality and morbidity, but also quality of life, ma'am, because that is also an
Yeah.
important factor to take into consideration.
Yeah, I understand. But for pre-marketing means the drug is not, been marketed. So you need a preclinical data, very robust. So you need a non-clinical data, that is animal data. And when you're doing a post-marketing data, you should have a preclinical data. You should have a drug trial in non-pregnant women. Okay. And you are not, and the drug should be safe, of course. And
Yes,
the
ma'am.
efficacy extrapolation is not adequate. So you're trying to do in that population of interest. See, efficacy extrapolation from non-pregnant woman to pregnant is not possible. So I want to do a study in pregnant women. It is the same principle which follows, like, when you talk about pediatric studies, we tell that, you have to do an adult and adolescent study and then only you can do in pediatrics. The same principle follows here. You have to have a non-clinical data, you have a non-pregnant data, and there is no safety over concern with us, and there is no efficacy extrapolation from non-pregnant to pregnant. Then only you can select a pregnant woman data. Okay. So another thing I
Yes,
wanted to
ma'am.
ask you, like, the regimen which is used here is 0, 1, 2, 4, 6. So is it the ideal regimen for hepatitis vaccine, which is given in
Uh,
India? What is the difference?
Ma'am, as far as I know, ma'am, this was so the general Thai immunization program states that there is 0, 1, 2, 4, 6. And
Uh-huh.
regarding Indian immunization program, if I remember correctly, ma'am, it is given at birth is given, then in a period of 2 months after it's given, then 4 months, and that is all I know, ma'am. So 2 months, 4 months, and then that can be a third dose given later on.
No, no,
But
0, 1, and 6 months is the Indian regimen. So this is a
zero,
totally different regimen. So again, this becomes a new drug. So
ha
when
ha
you
ha.
said, like, there is no asset in India, you can't simulate this design, because the regimen which is used in Thai is totally different, and we don't know
Yes,
what
ma'am.
is the Chinese people have used the regimen. So many times, when one country shows some drug trial which is not found to be efficacious, we don't try to do the same type of design again unless there is
Yes, ma'am.
controversial. So, for example, if Chinese data showed efficacy and Thai data showed non-efficacy, it means it was showed a negative result, then there is a controversial result, then you can do a third study. Or else in pregnant women, we don't allow this many studies. So, at least one study, and, you get into the data. Because these are
Yes, ma'am.
vulnerable population, there are a lot of ethical concerns which we are concerned with. For
Yes,
example, if
ma'am.
something goes wrong, so then the mother as well as the child, okay, and there are a lot of compensation issues, and if the child is disabled, born as disabled, for example, the sponsor has to pay liability throughout his life till the
Yes, ma'am.
child rectifies. So there are a lot of liability issues for sponsors. So sponsors will actually way off from doing a study in pregnancy.
Yes, ma'am.
So you have to understand this type of risk for the sponsor
Yes,
in
ma'am.
doing studies pregnancy. So they're not really motivated unless the need is so high. So for example, every single alternate patient of pregnancy is going to. So another thing I would ask, why more of cesarean section in China versus more of normal deliveries in Thailand? What do you feel? Is there any cultural difference there?
Ma'am, I think it's a cultural difference because in Thailand it's a bit more restrictive. As in women, like even in India, I would say that women are a bit more scared of cesarean sections because women feel that it is a much more involved procedure as compared to normal natural vaginal delivery. They say natural delivery is always better. That's what people think. And in China, ma'am, I don't have much knowledge about this, but from what I have read, in China's doctors have much greater agency over the decisions of the patient. Like consent is a bit shaky in very hospitals in China. So, and a cesarean section is a bit quicker in that it takes less amount of time. So people, so there's a higher incidence rate in China, ma'am. That is to the extent of my knowledge.
See, when you go to hospital for delivery in any setup, for example, in India also, normal deliveries are more preferred when it is home based. Okay, people will give a try, will try. See, there is indications for cesarean. Cesarean is not like for it's done for any case. Okay,
Yes,
if
ma'am.
there is some indication, then only go for cesarean. So there is a scientific indication. Okay, there are few gynecologists who are very unethical and they do cesarean for everything. That's a different story altogether. But if you see the data as such, so obstetrician, when the patient comes to hospital, the chance is 50% chance is cesarean. Okay. So what is the scenario in China is majority of the patients they go to hospital because the setting is so good and they are so
Mhm.
motivated, and the government has made so much of liability like everybody should come to hospital and delivery, and because of which cesarean sections are more. So in hospital setting in India also, if a patient gets admitted in a hospital, the chance is 50% with indication or without indications. And there are some hospitals prevalently in Bombay also, I can give you examples where 100% cesarean sections happen and they will tell you some risk, and patients then become ready for becoming cesarean section. So that
Yes,
is
ma'am.
one. Second, in Thai, half the people are not catering to hospital admissions. They're doing delivery
Mhm.
at home. And because of which the prevalence of normal deliveries is very much more. Unless it gets complicated and goes into hospital setting, then it lands up into cesarean section. Same like India. So
So, ma'am,
that,
would you say it's
yeah.
more of a infrastructure issue than a cultural issue? Because this was one
Yes,
doubt that
yes,
I had in my
yes,
mind.
yes. Infrastructural issue, because many times many of these Chinese things are mandatory. You are pregnant, you have
Mhm.
to come to hospital. They are communist in mentality as well as in their walk of life as well. So they are, everything is compulsion on the population. Okay, and that is not the thing in Thai, isn't it? Or for
Yes,
India
ma'am.
also, it's a democratic thing. So we have very lot of flexibility. I have choice of things. They don't have any choice. So I'll give you
Yes, ma'am.
one
That's why I
example.
mentioned consent first, ma'am.
Yes.
That's why I
So I'll
mentioned
give you
consent
one
was one
example
factor.
of, yes, yes. Consent is very blind because it's a communist country, no consent.
Yes, ma'am.
Okay, so you get a lot of data from China. So another thing, what is the issue here in China is China's all hospitals are accredited, and it is compulsion for all the hospitals to get accredited. Now you can't do these things in India. You understand? Even if you compel people way out, and people will not do it, and people will say why not voluntarily, and because there they have put systems in place. If you are accredited, you can put application for grant for government. You are possibly will get a grant. Yeah, this is that. This promotion is based on that. So everything is very streamlined. So people will follow guidelines. We don't have such guidelines. So if you see accreditation in Indian system, it is so skewed. No government hospitals are accredited, all private are accredited, and that is for business. You have to
Yes,
understand
ma'am.
that. So their objectives are totally different. So you have to understand the psychology of the governance as well as in the implementation issues. Okay. So another thing I wanted to ask Ansh, like you said in between that the one patient, the age was less than 18 years, and these were, and the one was stillborn, and there were twin deliveries. There are some data which,
Yes,
what
ma'am.
is this called as? Such data is called as in a
Uh,
protocol.
uh, I'm really sorry, ma'am. I'm not aware of the term.
So see, these are all protocol violations or deviations. So you're supposed to follow your
Okay.
protocol, and these people are not following. So some patient had a hepatic level high, they took the patient, and later they thought this is exclusion. Or else, what you can do, you have taken the patient already. You can write to ethics committee and you say like, give us a protocol waiver. By mistake, we took this patient. See, that fellow is already in the exclusion criteria. He's less than 18 years. You can't take that patient, isn't it? is
Yes,
not
ma'am.
fitting in your inclusion criteria. So this is called as protocol violation. Other way around, you are not followed your protocol, and that is actually been submitted. And many times CC takes a lot of action on investigators. And if you want to continue with that patient because you are losing own data, you can put for protocol waiver, and that is again acceptable. But you have to have a scientific logic for that, and ethics committee will take its own call for allowing it or not allowing it based on the evidence. Okay.
Yes, yes, ma'am.
Yeah.
Ma'am, regarding
So
this, regarding the ethics that you mentioned, so that was multicentric trial. No? So there were 17 hospitals involved. So each hospital's ethics committee did approve this trial, but as you said, there was not very significant mention. Like they have mentioned that there have been some protocol violations, but they didn't mention what they did to rectify those protocol violations. So that may be also a
so that
limitation of
they
the study,
don't
ma'am.
write in publication, but at local level they may have done because in data they have mentioned that thing, isn't it?
Yes, ma'am.
Yeah. So are there any ethical challenges in doing studies in pregnancy? What are the ethical challenges? For example, I am in a trial and I'm not supposed to become pregnant. If I become pregnant, what happens?
So, ma'am, the biggest thing, as you said, if it's a trial which is geared for non-pregnant individuals, so the biggest thing is the exclusion criteria would be pregnancy. So you're automatically excluded from the trial. So that
No,
is
I
the
become
biggest
by
thing.
mistake. I become pregnant. I'm not supposed to become. See, that is what is protocol violation. Na? I'm not following your protocol. I'm not used my contraceptives properly. There is double contraceptives used in to prevent pregnancy. And I'm not a participant of the program. My husband is a participant, and I'm not supposed to become pregnant. But I became now. What happens?
So, ma'am, even whatever might be the reason behind being pregnancy, whether it be accidental or intentional, according to the study protocol, if there is mentioned that there is exclusion criteria, so even if I take data of yours, ma'am, even if I take data till the end, I have to exclude the data from the analysis at least, so that it doesn't affect the results of the study and your confidentiality isn't compromised there, ma'am.
So it is not that simple. That is what I'm saying. So I'm not supposed to become pregnant. I became pregnant. So that pregnant woman, you give choice. Are you going to terminate because the risk of abnormality is this much? And if she doesn't want to, get into this abortion thing, so then she is continued and she's followed up till 1 year of delivery. And because till 1 year of delivery, you will understand the, whatever congenital anomalies will come into existence. Until then, the PI has to follow up that patient because patient is not willing for abortion. Okay. And that taking care of that patient is investigators and sponsors responsibility or liability. So only we say that your instructions in the clinical trials should be very clear. Patients are so illiterate, they will not take into hindrance. And when you're taking a fertile age group, you should be very much clear in your instructions. That is very important. I'll give you one example of our own trial. We had brivaracetam study in neurology, and the male was an epileptic patient. His wife was not supposed to become pregnant, and there were 5 patients who became pregnant in that study, and they were not ready to abort. Okay, and then we had to follow that patient till 1 year of delivery. And incidentally, there were no abnormalities in the babies, but we had to follow 3 monthly scans of the babies and in the pregnancy, and later after delivery also we had to see till 1 year the pediatrician certification that there is no congenital anomalies. So there's a lot of task for the for ethics committee to follow up these patients because the patient was in clinical trial and his wife was not supposed to become pregnant. She's not a participant. Don't understand that. And there the thing, arrives so this is difficulty. You are a participant, then it is very easy to handle. But when you're not a participant, then becomes very risky. So pregnancy itself is a risky ball game for any clinical trial setting. And you as a clinician has to know that what are the ethical challenges. And every time you have to consult obstetrician because they are the experts. You can't say that I'm the expert, I know everything. No. Even if I know everything about the drug, I don't know what can happen in pregnancy. So I have to take all my, directions, scientific value additions from a gynecologist or obstetrician who is an expert in that field. And in that team, he has to be there, whatever study you do. And that will be a scientific requirement of the study. Okay.
Yes, ma'am.
Thank you very much. You did a very good job, Ansh.
Thank you so much, ma'am. If the audience has any questions.
Yeah, that is what I'm asking. Audience have to ask a lot of questions. Experts asking questions is a different this thing. Prashant, you should ask questions because he is your competitor, isn't it? You read about RCTs, isn't it? You should have a lot of questions. Vidushi, you only doing a coordination part is not enough. You have to ask questions, isn't it?
Yes, ma'am.
So no questions. Then we are finished with it. Both have done a good job. That was a very good learning experience at MBBS level. You have done a marvelous job. And I think you will become good researchers ahead and a good clinician.
Thank you so much, ma'am.
Thank you.
Yeah.
Ma'am, before we proceed,
Yeah.
I would request everybody to please switch on their cameras if possible so that we can take a picture.
Photo घेतो मुले. Everybody else also has to put on their cameras. Krish, Ajmera, Shruti Tilak, Disha Agarwal, they are there or not? Or they are only kept their thing and they have left.
Okay, ma'am. I think we are done.
Yeah, thank you very much. So can I leave, Vidushi?
Uh, yes, ma'am. Thank you for joining us and for your insightful reviews. Thank you so
Yeah.
much, ma'am.
Yeah.
So with this, we come to the end of the journal club. I would like to thank the speakers, Shruti, Prashant, and Ansh for their efforts in delivering meticulous presentations. I would want to extend my generous thanks to our audience. Your participation made this club even more interesting. Finally, I would like to thank the organizing committee of Aspire for making this event possible. Stay safe, keep researching. Thank you.
Thank you and thank you very much and bye-bye.